My guest today, again, is Rob Louw.
Today I'm going to talk about photosynthesis, the process that basically drives all life on Earth. Now, I've got quite a few slides to get through, so I'm going to start off right away and go through my slides as best I can in the time ahead. Now, photosynthesis is the process by which plants use sunlight to convert CO2 and water into glucose, which is the building block of cellulose, and the process produces oxygen, as shown by this chemical reaction you see here on the screen. In the first few slides, I'm going to look at historical levels of atmospheric CO2, and then in the next few, we'll look at how important photosynthesis is to the whole food chain. And then this will be followed by a review of a plant leaf's anatomy. And then I plan to dig into more details about the photosynthetic process and discover how plants have cleverly adapted to a particularly changing carbon dioxide environment. Let's have a first look at carbon dioxide over the past 600 million years. As you can see from here, if you start at the left-hand side of this graph, roughly 600 million years ago, carbon dioxide was at considerably higher levels than it is today, some 8,000 parts per million. And ever since then, it's been steadily in decline to where it's reached today's levels of about 427 parts per million. So we can see that carbon dioxide levels have been changing a lot over Earth's history. I'm going to dig into this in a little bit more detail and give short explanations of why we see these changes in the levels of carbon dioxide. And let's begin with the Cambrian. At that time, no plants existed to draw down CO2, and normal volcanic and seafloor spreading processes kept adding CO2 to the atmosphere. And with no biological weathering on land, CO2 remained high, making Earth warm while the continents were still barren. This was then followed by the Ordovician period, where carbon dioxide started to fall because shallow seas and rapidly evolving marine life removed carbon faster than volcanoes at the time could replace it. And so remember, the Earth was a very volcanic entity during that time. Vast limestone deposits formed, and early calcifying plankton locked carbon into shells, while new mountains weathered quickly, and all of these processes consumed CO2. The resulting cooling culminated in an ice age that reinforced the CO2 decline. Bear in mind, cooler seas have an ability to absorb more carbon dioxide than warmer seas do. We then move on to the Devonian period, which is when land plants and later deep-rooted trees evolved, greatly accelerating rock weathering and removing CO2 from the atmosphere. Large amounts of organic carbon were buried in developing soils, sharply reducing CO2 levels. This was a major CO2 reducing event, paving the way for even bigger carboniferous coal deposits that followed. And this takes us into the Carboniferous area, where carbon dioxide levels really did drop fast. Vast tropical swamp forests accumulated huge quantities of plant material, much of which became buried and preserved as coal. This long-term carbon burial drove down carbon dioxide levels sharply, and eventually approaching near modern values. And it was one of the most important CO2 reducing events in Earth's history. So it's the early evolution of trees and plants that really sucked a lot of the carbon dioxide out of the air. We then move on to the Permian period, where carbon dioxide began to levels began to grow again. And this was because of white rot fungi, which evolved, and they developed an ability to decompose lignin. This greatly reduced the long-term burial of woody material, weakening the strong CO2 drawn down that had characterized the Devonian and Carboniferous periods. So what happened is the carbon dioxide wasn't being removed as fast as it had been prior to this event. Then along came the Siberian traps, which is about 252 million years ago, when one of Earth's largest volcanic events occurred, releasing vast amounts of carbon dioxide and other gases into the atmosphere. And this triggered the end Permian mass extinction, which is the largest extinction event to have occurred in Earth's history. Later, we reached the Triassic, Jurassic, and Cretaceous period, which is 250 to 65 million years ago. And this happened as Pangaea broke apart. Widespread volcanism and rapid seafloor spreading released large amounts of CO2, contributing to warm global climates. The levels there rose nearly back to 3,000 parts per million. In the oceans, plankton increasingly formed calcium carbonate shells and organic-rich sediments, bearing carbon and partly offsetting volcanic CO2 inputs. Then periodic ocean anoxic events in the Cretaceous led to especially high rates of organic carbon burial. Getting closer to modern times, in the last 50 million years, the rising Himalayas increased weathering, grasslands expanded and formed new swells, and cooling oceans absorbed more carbon dioxide, gradually lowering atmospheric CO2 towards modern levels. And then in the more recent past, we saw ice ages developing, and consequently CO2 levels dropped to about 180 parts per million, just 30 parts above the level where C3 plants begin to fail. C3, by C3 I mean photosynthetic pathway, which we are going to go into in a lot more detail later on in my presentation. And during this period, Earth came surprisingly close to starvation point for vegetation. Had that happened, we may well not even be here today. But at today's 427 parts per million, we are far below the long-term geological norm, which is about 2,500 parts per million. So we got a long way to go back to get to the geological average. But it should be remembered once again that below 150 ppm, plant life is almost impossible. Okay, so I think that sets the scene, and we can move on to answering the question, why did life choose CO2? And the reason is that billions of years ago, the atmosphere and oceans were rich in CO2, but had almost no organic carbon and no oxygen. To build sugars, proteins, and cell walls, early microbes had to use CO2. There was no other. There was no other compound in the kind of abundance that CO2 was. Life used CO2 because nothing else was available in quantity. Long before plants existed, ancient bacteria evolved ways to turn CO2 into useful organic molecules. So this goes right back billions of years when this was happening. And during that time, before plants even came into existence, several carbon fixation pathways existed, the Calvin cycle, and we're going to learn more about that, reverse TCA, and others. These pathways appeared hundreds of millions of years before chlorophyll or leaves. And interestingly, plants gain photosynthesis by absorbing ancient cyanobacteria, which became the chloroplasts in the leaves. And the cyanobacteria already had ribisco, a key enzyme in the Calvin cycle, and they were already using the Calvin cycle. Now, photosynthesis provided energy from sunlight to reduce carbon dioxide efficiently. And plants use CO2 because their cyanobacterial ancestors already mastered the chemistry. But plants kept it and perfected the machinery. So our plants were smart. They didn't reinvent the Calvin cycle or photosynthesis. They just took what the cyanobacterial ancestors had done and perfected it for today. All right, this brings us on to the food chain and how plants power all life. Plants and phytoplankton sit at the base of almost all food chains, and because they cannot move to hunt, they must make their own food using sunlight, water, and carbon dioxide. And then through the process of photosynthesis, they convert solar energy into energy-rich sugars. And these photosynthetic organisms are known colloquially as primary producers. Herbivores on land and zooplankton in the ocean feed on these primary producers, and then in turn, small predators feed on the herbivores and zooplankton. So we move up the chain and we find that larger predators feed on smaller ones, passing the originally captured solar energy up the chain. Humans as omnivores and top predators sit near the apex of both terrestrial and marine food chains. But in the deep ocean, where no sunlight reaches the seafloor, deep sea life is actually still supported by photosynthesis by what we call marine slow, which is a slow rain of organic particles produced by photosynthesis at the surface. So what is happening? They're getting their food supply from the photosynthetic processes that have had closer to the surface of the sea. But there is a rare exemption on Earth where sunlight can't drive the photosynthesis, and this is where bacteria use chemosynthesis at the Earth's hydrothermal vents. And in the chemosynthetic process, chemical energy, not sunlight, is used to support their unique ecosystems. So in summary, the sun's energy is the energy source from which plants derive their energy to grow. Likewise with phytoplankton, this then leads to herbivores and zooplankton, which are predated on by predators, and eventually it gets to humans as the apex predator. And with deep sea vents providing the only major non-solar food chain on Earth, and only in the very deep sea areas, we don't benefit ourselves from that. Now, photosynthesis on land occurs in the leaves of plants. So what I'd like to do is take a look at the leaf's anatomy to see where the action takes place. And here's a diagram of a plant leaf. You'll notice that it has an upper and a lower epidermis to protect the various cells that are inside the leaf. You have leaf hairs on the surface, and you have two groups of cells inside. You have the palisade mesophyll and the spongy mesophyll. And I'll be taking a bit of a closer look at what these different elements of the plant are. The first one, if we look at these little tubules here, this is where this is called phlegm, and this transports food from the leaves to the rest of the plant. So the food that the plant is making, that the leaf is making, is transported by the phlegm to feed the rest of the plant. At the same time, xylem brings water up from the roots and minerals from the roots to feed the leaf and help it develop its full leaf structure. We then move on to this vascular bundle, which is made of the phlegm and the xylem, and it's protected by the bundle sheath cells. So this is the passageway for nutrients to get to the leaf and for products that are made in the leaf to be transported back to the rest of the plant. Then in addition, we have this is a very important part of the plant. It's the stoma. It's an opening in the underside typically on the underside of a leaf. And the stoma are protected by guard cells which can open and close them depending on the leaf's needs. The chloroplasts sit inside these mesophyll cells, and this is where the action takes place. This is where photosynthesis happens and where the plant sugars are manufactured. As I said, stoma are a very important part of the leaf because they play a key role in exchanging CO2, oxygen, and water with ambient air, and the chlorophyll itself sits inside what we call the thylakoid membranes, which in turn are inside the chloroplasts. So next in this presentation, I'll be going into, I'll come back to this later and look at it in a bit more detail. But let's first start with the basic part of the important basic element of a leaf. And those are the leaf stomata. They're small pores on the leaf surface that play a vital role in gas exchange. And through the stomata, plants take in CO2 for photosynthesis and release oxygen as a byproduct of that process. But stomata also allow water vapor to escape, transpiration, which can lead to water loss under dry condition, but also helps to cool the leaf and draw nutrients upwards. There is a problem with if the stomata allow too much water to evaporate, then the plant will suffer. The number and opening of stomata adapt to changing environmental conditions and especially carbon dioxide levels. And here's a microscopic image of the leaf stomata. Incidentally, these are quite easy to find. I was showing this presentation to a young nephew of mine, a 10-year-old nephew, who had just been given a microscope as a birthday present, and I went and picked a few leaves off our rose bushes and put it under his microscope, and lo and behold, there they were. So he believed what I was telling him. Anyway, each pore is flanked by two guard cells that control its opening and closing. This is the guard cells on there. There's the stomata there, and there's the guard cells sitting around the outside. Now, their density varies with atmospheric CO2 concentration. So the more CO2 there is, the fewer holes there are. The less CO2 there is, the more holes there are, because it's more difficult to get the carbon dioxide into the leaf. So higher CO2 means fewer stomata and lower water loss. Okay, so let's see how the density of these stomata vary under the conditions of a controlled experiment. And here we have it. These are experimental observations that were made under controlled conditions using 5 plant species at different carbon dioxide levels over a period of 3 weeks. And you can see on this graph here, you've got the relative change in stomatal density on the vertical axis, and on the horizontal axis, you have the carbon dioxide mole fraction. And as you can see from the diagram, each rise in carbon dioxide produces a measurable a drop in stomatal density. If you go back, if you take carbon dioxide concentrations down here to about 220 parts per million, and you then raise it up to nearly 350, you can see this line slopes down quite steeply. And this happens immediately. Plants are growing in a richer environment. It doesn't take time to develop. These plants were only 3 weeks old. So the question is, does the same happen in nature? And to answer that, I'd just like to take a quick look at what has happened to carbon dioxide in the recent past. If you go back to the mid-1700s, carbon dioxide has risen from a level of about 280 parts per million to 425 over the past 360 years. And given what we've just seen, we should ask ourselves, what effect might this have on the structure of leaves? We've seen that in an experimental environment, the number of stomata on the leaves reduces quite sharply as the carbon dioxide levels increase. And here we have it. Over the past 200 years, stomatal density has fallen by nearly 40%, which is clear evidence that plants have adapted to rising CO2 levels. Here's another, this graph I've got here is again, the vertical axis is a relative change in stomatal density expressed as a percentage. And on the horizontal axis, you have time. These are years on the horizontal axis. But of course, they equate to differing levels of carbon dioxide. And if you go back to the pre-industrial area where values of carbon dioxide were 280 parts per million, and you take it up to the year 2000 where it was closer to 400 parts per million, you can see once again that there's quite a marked decline in the number of stomata. Now, it turns out that this the change in leaf stomatal density is a very useful paleontological tool. If you can carbon date leaves from a known species over many years, hundreds if not thousands, you can then examine them, and you can look back and count the number of stomata per square centimeter, or whatever unit you choose, and you can compare that to modern day levels, and from that you can deduce what the level of carbon dioxide in the atmosphere would be. It's one of the most accurate ways of determining historical CO2 levels, because we didn't have the instrumentation and the methods around to measure carbon dioxide. It's only in recent times that we've been able to do it analytically. So it's a very useful tool in the toolbox of paleontologists. Okay. So what are the major factors influencing photosynthesis? Firstly, there has to be adequate sunlight, chlorophyll, water, and minerals to allow photosynthesis to run faster. Higher CO2 concentrations boost plant growth up to an optimal level. Higher temperatures also boost temperatures again up to an optimal level. Yes, the plants grow with more and more carbon dioxide, but there comes a limit where adding more doesn't give you much further benefit. Similarly, if you give your plants too much water, you could end up drowning them. So there's actually an optimal point at which you want to grow them. But this is an important point. To plants, carbon dioxide is not a pollutant. It's the raw material for making sugars. In fact, if there was no carbon dioxide, or dare I say, if it went below 150 parts per million, life on Earth would not exist. So it's a vital ingredient as far as plants and consequently us humans are concerned. Now, commercial growers are aware of plants liking carbon dioxide, and so they use it to enrich their yields, as you can see in this image. And this is a CO2 generator which converts natural gas into carbon dioxide, and that greatly enhances the plant growth and productivity in greenhouses. And greenhouse growers add as much CO2 as is economically feasible, up to about 1,300 parts per million. They don't get the carbon dioxide for free, even though in many chemical industries it's actually a waste product. But anyway, they've got to pay for it, and you add it to the point where you get to an economic break even, where the cost to put more in doesn't generate greater yields. So the optimum seems to be roughly at 1,300 parts per million, which is roughly 3 times the level, that we have today. So more CO2 suppresses harmful photorespiration, a process which we'll discuss in a bit more detail, which is why greenhouse crops thrive when CO2 is enriched. The more CO2, the less water the plants need. And this is not an issue, obviously, in a greenhouse, but it makes a big difference in the natural world. Plants growing in arid or semi-arid regions benefit enormously through increased carbon dioxide levels, because they then require less moisture, and given that it's in short supply, it's beneficial to the plant. Now, doubling carbon dioxide roughly halves a plant's water requirement, making it far more drought resistant. So we know that growers deliberately add CO2 to boost yields, but just how universal is that effect? So let's look again at what happens with controlled experiments, where this time we're going to have a look and see how much a plant grows as we change carbon dioxide, rather than how many stomata there are in the leaves of the plants. And here's the first experiment. The plant on the side is the common velvet leaf, grown at different carbon dioxide levels. All the plants are of the same age and grown under identical water, light, and nutrient conditions. And at the left is a sort of starvation level close to the last glacial maximum, which happened 27 to 18,000 years ago, when carbon dioxide levels fell as low as 140 parts per million. And at the right are future growing conditions, which we'll get to in the next century, where carbon dioxide levels are 700 ppm. And as you can see, there's a pretty strong response. 700 ppm produces much, much more growth than 150 ppm does. Now, the same experiment has been conducted with pine trees. You've got 4 images here. The pine on the left was grown at ambient conditions. This is back in 1994, when it was 385 parts per million. And then it was the concentrations were increased by 150, 300, and 450 parts per million, and the trees were grown for 2 years. And again, as you can see, the tree at the ambient level is considerably smaller than the tree that was grown at 835 parts per million. So as the carbon dioxide levels increase, so the size of the trees, or the rate at which they're growing, increases as well. And towards the end of the presentation, I'll show you a NASA photograph which confirms the greening effect, and it's greening the whole of the Earth. I think it's a fact which is not that widely known. All right. Now, clearly, plant growth increases dramatically with higher CO2 levels, and this explains why the Earth is greening. Over the past 200 years, it's risen from 280 parts per million to 427, which is today's level. But today's photosynthesis is still limited due to current ambient levels of CO2. The previous slide, I think, illustrated that pretty clearly. So the same pattern of enhanced growth with higher CO2 is seen in all photosynthetic organisms, forests, crops, phytoplankton, all of which we rely on for food and oxygen. Okay. So much so about carbon dioxide. I did mention that there was actually an optimum level, but before you even get there, you can't just give your plant lots of carbon dioxide because it doesn't depend on that only. It's a key driver of the photosynthetic process, but in order to maximize the benefits of more carbon dioxide, you've got to add other nutrients to the plant. And the key concept of fertility is Liebig's law of the minimum. And for true growth, plants need mineral nutrients: nitrates, phosphates, potassium, magnesium, calcium, phosphorus, and trace elements. And they are needed to build the proteins, chlorophyll, and cell structures from the sugars produced by photosynthesis. Now, in modern agriculture, the suite of nutrients are mostly provided by fertilizers made from fossil fuels. An important point which I think is often lost on people today is just how important fertilizers are. If we were to try and do away with them, then we would have tremendous difficulty producing the food we need to feed the planet today. And yes, Liebig's barrel, which explains the concept that he came up with, and he stated that a plant's potential is limited if even one growth factor is deficient or missing. And what I have here is a barrel with vertical wooden slats cut to different lengths, and each slat corresponds to one of the macronutrients that the plant needs, from sulfur to nitrogen to potassium to carbon dioxide. Now, in addition to that, we also have micronutrients: chlorine, iodine, manganese, iron, zinc, molybdenum, and what have you. And all of these should actually have their own slat. And what this illustration is the message this illustration is conveying is that if you were to fill this vat with water, you could only fill it up to the level of the shortest slat. And after that, you would have lost yield potential. So, yes, you could make carbon dioxide a lot richer and make its slat sort of go right up to somewhere there, but it would make no difference, because if this particular, the slat that is the rate determining nutrient is in short supply, then the plant won't benefit. So this needs to be considered. There are a whole host of additives that a plant needs for healthy growth. And in addition to these slats, plants also need the correct soil. You wouldn't try and grow your plants in, for example, beach sand, but fertile agricultural fields, that's great. And temperature. If it's too cold, plants don't grow, and if it's too hot, they don't either. Now, I'd just like to draw attention to one little thing here, and that is that paleontologists often use tree rings to reflect historical temperatures. But as you can see from this example, this can be quite misleading. Ring growth is strongly influenced by moisture, CO2 availability, soil nutrients, disease, and local conditions. And this makes rings an uncertain and, quite frankly, an unreliable paleoclimate temperature proxy. However, it is frequently used in the literature. And I would just say, when you are reading the literature, take some caution. Check whether the claims that are being made can be validated. But that's more or less as an aside. So what's happening to plant growth now that we are recovering a bit in our carbon dioxide environment? As I've said several times, for tens of millions of years, one of the most important plant nutrients has been falling below the optimum concentration for plant growth. But now that they're beginning to the CO2 is beginning to recover to more optimum levels, other nutrients will have to increase to sustain more vigorous growth of plants, and which is pretty obvious from the slide which I've just shown you. I'm going to dig a little bit deeper now into the actual what's happening in photosynthesis. And what's happened is the three photosynthetic pathways have evolved over time, basically learning to cope with changing environmental conditions. And I'm going to go through these three now. First of all, let's just take a high-level view of photosynthesis. And as I mentioned in the anatomy slide earlier on, photosynthesis takes place inside the chloroplasts, which sit inside the plant cells. Oops. The chlorophyll itself, the compound I showed in the first reaction, the first slide, sits in the thylakoid cells. It absorbs sunlight, it excites electrons, splits the water, releases oxygen, where you can see down here, it releases it there, and powers ATP and NADPH formation. That chlorophyll, obviously, it needs sunlight and it needs water in order to for that reaction to take place. The chlorophyll absorbs light mainly in the blue and red parts of the visible spectrum. But as we all know, it absorbs very little green light, which is why plants appear green. They reflect it. The light in the middle of the visible spectrum is not absorbed by chlorophyll, which is why most leaves are actually green. Now, ATP is the cell's energy currency, which we see here. And this is produced in the light reactions that are occurring in the thylakoid cells. And this produces the power to drive what we call the Calvin cycle over here. After this slide, I'll be digging more into what's going on in the Calvin cycle. NADPH is a high energy electron carrier, which is delivering power, electron in the form of electrons and hydrogen, to build sugars in the Calvin cycle. So photosynthesis starts reactions here, generates ATP and NADP, which then go into the Calvin cycle. And what then happens after the Calvin cycle, you get NADP plus, and that's an empty electron carrier, which is returned from the Calvin cycle to be re-energized into NADPH. And so the cycle goes round and round inside the chloroplast. And one last thing, ADP plus pi is a low energy form of ATP, this energy up here, which is recycled in the light reactions to make fresh ATP using sunlight. So you can see this, the sunlight is driving the light reactions, producing the compounds that you need for the Calvin cycle, which is producing the glucose, as you can see here. And then the spent compounds are returned to the light action part. I'll summarize it for you in 3 simple steps. Chlorophyll captures the sunlight, and the light is absorbed by chlorophyll in the thylakoids, and this energy splits the water, releasing oxygen and creating the energy carriers ATP and NADPH. Energy is transferred. ATP and NADPH move from the thylakoids into the stroma, carrying the sunlight energy with them. And then CO2 is turned into sugar. And in the Calvin cycle, carbon dioxide is combined with ATP and NADPH to make glucose. And the used energy carriers return to the thylakoids to be recharged. So it's almost like a car battery, which you charge and discharge and recharge. It's going around and around in a cycle like that. So how did the evolution of these 3 cycles come about? The Calvin cycle itself, there are 3 versions of it, but we need to go back in time to see where key developments took place. And the first one happened almost 3 billion years ago when the enzyme rubisco evolved. But at that time, carbon dioxide was greater than 10,000 parts per million. But now it's used in all these photosynthetic pathways. The simplest and the most abundant pathway is called the C3 pathway, and it's used by 85% of plants, such as trees, grasses, wheat, rice, and it evolved 400 million years ago when CO2 concentrations were between 2,000 and 4,000 parts per million. The next pathway to evolve was what we call the C4 pathway, which is efficient in hot, bright, sunny conditions. And it's what the maize, sugarcane, and sorghum plants use. That evolved about 30 million years ago when carbon dioxide was much less, about 400 parts per million. And we're going to see why this is so important, because as the carbon dioxide levels were dropping, so plant productivity started to decrease, and a different photosynthetic pathway was needed to keep plant growth rates up at lower carbon dioxide levels. And then another clever pathway came along called the CAM pathway, and this is used by desert plants such as succulents and cacti, and it's a very clever way of conserving water, and they evolved 20 to 25 million years ago when carbon dioxide itself was very low, 200 to 300 parts per million. And if you recall from the previous slides I showed you, when carbon dioxide levels are low, the leaf stomata count is high, and when the stomata count is high, the plant loses a lot of water due to evaporation. And in arid conditions where water is in short supply, that's the last thing that a plant needs. But the CAM pathway has worked a clever way to get around that. And each of these represents a brilliant evolutionary response to declining CO2 and rising O2. I mean, in a nutshell, oxygen is actually a poison for plants. It's essential for us, but as far as plants are concerned, oxygen is something they would prefer not to have. So let's take a brief look here at the C3 pathway. This is we're looking inside the mesophyll cells. And this is where the rubisco enzyme resides and where the Calvin-Benson cycle takes place. The plant takes in carbon dioxide through its stomata into the rubisco, grabs that and uses it for the cycle. And the oxygen that's produced in the process, the is then expelled through the stomata, and some water is also lost through those holes. And this is the most common pathway, and I'm going to take you through a series of steps which will describe what happens here. So the C3 plants fix carbon dioxide, which they've taken in here, with rubisco mesophyll cells, but because rubisco is exposed to oxygen, photorespiration becomes a major energy loss in hot or dry conditions. CO2 and O2 both enter the leaf through the stomata. You also get oxygen, some oxygen coming in through the stomata, but most of it is generated in the plant. And mesophyll cells are directly exposed to the air. And this means rubisco sits in a mixture of both carbon dioxide and oxygen. Okay, so that becomes a problem for rubisco, because it likes carbon dioxide. It also likes oxygen, but oxygen produces bad stuff for the plant. Under the ideal cool, moist conditions, CO2 levels are high enough for rubisco to function normally. And it fixes the carbon dioxide directly in the mesophyll cells. What then happens is that the rubisco attaches carbon dioxide to a 5-carbon compound, which is RUBP, forming a 3-compound product, hence the name C3. That's where the C3 pathway gets its name from. This then feeds straight into the Calvin-Benson cycle, producing sugars using ATP and NADHP from the light reactions. We saw that in an earlier slide. But rubisco has a flaw. It also reacts with oxygen when it binds O2 instead of CO2. A process known as photorespiration occurs. And in this process, carbon is wasted, energy is wasted, and no sugar is produced. And this is worse in hot, bright, dry, sunny conditions because the stomata close, reducing CO2 intake. The stomata close to conserve water. That reduces the carbon dioxide intake, but the oxygen levels remain high, and that's not a good position to be in. So in summary, the Calvin-Benson cycle works best under cool, moist conditions and moderate light. It's used in all 3 synthetic pathways, and we've still got to examine the other 2. The cycle was discovered in the 1940s and 50s by Melvin Calvin, Andrew Benson, and James Bassam. And in fact, Calvin was awarded the Nobel Prize in Chemistry in 1961. So it's only for the past 60 or 70 years or so that we've actually really known how the photosynthetic process in plants actually functions. The C3 pathway, as I've said several times, has a problem, and the 2 problems are, 1, photorespiration, which is toxic to the plant, and water loss. Unfortunately, rubisco does react with oxygen, causing the photorespiration, which wastes energy and releases CO2. And this increases when it's hot and dry, or when CO2 levels are low, reducing the photosynthetic efficiency. And byproducts of this process are ammonia and hydrogen peroxide. I've mentioned when Rubisco first evolved, it was highly efficient as a pure carboxylase. But when oxygenetic photosynthesis by cyanobacteria began enriching the atmosphere with oxygen, this was the great oxygen event about 2.4 billion years ago, a design flaw in Rubisco became apparent. And its flaw is that it can also react with oxygen to give photorespiration, which wastes energy and carbon. And here's an image of the Rubisco molecule. As you see, it's a pretty complex enzyme, and it's capable of catalyzing the carboxylation reaction or the oxygenation reaction. Now, carboxylation is good and desirable, but oxygenation is bad and undesirable. What happens in carboxylation is catalysis of carbon dioxide is the first step in the Calvin cycle, but with oxygenation, the Rubisco binds with oxygen, which gives rise to photorespiration and wastes the plant energy. And because of this inefficiency that developed, this resulted in the evolution of the what we call the C4 and CAM photosynthetic pathways. Now, instead of evolving into a perfectly selective enzyme, Rubisco became highly abundant to compensate. Different forms evolved in bacteria, archaea, and plants, but all share the same basic chemistry. Now, in some plants, Rubisco is now housed inside chloroplasts and supported by carbon concentrating mechanisms like C4 to reduce its wasteful oxygenase activity. And in short, it's an ancient enzyme well adapted to high CO2 world, but whose inefficiencies in today's oxygen-rich atmosphere have shaped evolution of diverse photosynthetic strategies. Oh, sorry, this is a repeat of the previous slide. Here we go. Let's have a look at the C4 pathway. And this is the efficient pathway, and as you can see, the carbon dioxide doesn't go straight into the Rubisco. In such a cell, the plant is acting cleverly to keep oxygen away from the Rubisco and pump in carbon dioxide so that it reacts with it. And here are the steps. They physically separate carbon dioxide from the Calvin cycle and then pump concentrated carbon dioxide directly into the Rubisco in this little process that happens on the side here. Two different cell types work together. The mesophyll cells take in carbon dioxide from the air. The bundle sheath cells house Rubisco deep inside the leaf where O2 levels are low. And carbon dioxide is pre-captured by PEP carboxylase, which grabs the CO2 efficiently and without interference from oxygen. And then it turns the CO2 into a 4-carbon acid, malate or aspartate. And that's why this is called the C4 pathway. So what the plant is doing is it's capturing the carbon dioxide, it's concentrating it, and then pumping it across to the Calvin-Benson cycle so that Rubisco sees more carbon dioxide, certainly, than it sees oxygen. The 4-carbon acid is transported to the bundle sheath, and inside the bundle sheath, the acid releases carbon dioxide right next to the Rubisco. and CO2 builds up to very high concentrations. And high CO2 plus low oxygen means that Rubisco behaves properly, and it sees almost pure carbon dioxide in the cell, which then means that it performs carboxylation and very little oxidation. So photorespiration is almost eliminated, and the Calvin cycle runs more efficiently. And last but not least, we come to CAM photosynthesis, which is what happens when it's too dry, too hot and too dry for even 3-4 plants. And what happens in this process is that plants close their stomata completely during the day to prevent water loss. They capture carbon dioxide at night when it is cooler and more humid. And at night, the stomata open, CO2 fixed by PEP, carboxylase, and stored as malic acid in vacuoles. And then during the day, the stomata are closed. CO2 is released internally and used in the Calvin-Benson cycle. So it's a very clever plant. It's learned to capture carbon dioxide at night. It can't use it at night because there's no sunlight, and so there's no energy for the reactions to start. But as soon as the sun rises, it closes its stomata. It's now got its store of carbon dioxide inside the leaf, and during the hours of daylight, it releases the carbon dioxide into the Calvin-Benson cycle, and so the plant growth continues. Here is an example of one that's called a speckboom in South Africa. It's also known as the elephant bush or a dwarf jade plant, and you can see there it's growing quite well in pretty arid conditions. This is, as I said, it's the CAM process, and it greatly reduces water loss and is ideal for desert plants and succulents. Now that we understand the way in which plants use CO2 to their and our benefit, let's consider the effects of the recently rising CO2 levels, which I'm pretty sure we're all aware about. We've heard much about rising carbon dioxide, but let's see what that is doing to plant life. Now, the first thing to look at is the Keeling curve, which has been recorded back since the early 19th. I think it was started in 1959 by a guy named Keeling. There's an observatory on one of the Hawaiian islands, which incidentally is a volcanically active island, so active that an eruption 3 years ago, so much lava poured out that it's blocked the road to the observatory, and you can only reach it by helicopter now. Anyway, it's been chosen as a place where you monitor the Earth's carbon dioxide levels. Fortunately, carbon dioxide is a gas that mixes very well, so one has to assume that it's a fairly representative sample. And it's good quality instrumentation. So we have a very good record of carbon dioxide, certainly since 1960. Prior to those years, we've had to use proxies to work out. Well, there've been some measurements that were taken in other laboratories, but going back hundreds of years, we've had to use proxies to construct the charts that you saw earlier on. You'll notice that there's a zigzag in this chart, and that is because most of the Earth's greenery is in the northern hemisphere, and this is the effect of plants losing their leaves in winter. Deciduous plants are responsible for that zigzag in the curve. So that at the end of end of autumn, they drop their leaves. Carbon dioxide levels can drop accordingly, and then increase accordingly, and But then at the end of sort of getting towards springtime, leaves start growing again, and the carbon dioxide gets sucked out of the air. So there's the carbon dioxide rising because of fewer leaves, and there it's dropping because of more leaves and photosynthesis taking place. Yeah, in some ways it's good news that our carbon dioxide levels are rising, because in fact we're still in a low carbon dioxide environment compared to most of Earth's history. Yeah, you can see carbon dioxide is rising at about 2.5 parts per million per annum, which means that if we carry on at this rate, it'll double to about 850 parts per million at the end of the next century. But look what's happened to global food production compared to population and land use. It's been an absolute productivity revolution. The top curve here is look how crop yields have increased over recent times, and particularly if you look from about the 1960s onwards. If you consider that our food production yield was at 100 then, we're at about 350 today. It's an incredible improvement. At the same time, the amount of land that is being used to produce those crops has hardly increased at all. So what this means is that we're getting massively more efficient crop yields or production, which for the time being at least is comfortable in terms of feeding a rising global population, which you see on this curve here. So fortunately, as things are at the moment, the crop yields have increased because of the CO2 fertilization, which you saw in the rising CO2 in the previous chart. And of course, then nitrogen fertilizer use and other nutrients are clearly needed to do this. And quite frankly, this rise in CO2 has coincided with what is not often talked about, but one of humanity's greatest successes stories, and that's the surge in plant food production with less land. And we can see that more clearly in these charts that I'll show you. You've got on the vertical axis the production in millions of tons, although each of them is slightly different because depending on the crop. But you can see here there are 4 crops on this slide. All of them have been increasing quite significantly over the past 60 to 70 years. And here we have sugarcane, rice, potato, and soybean. They're all showing the same patterns. So clearly we are benefiting from it. Improved fertilization, greater carbon dioxide levels have helped us to produce the more food that we need to feed an ever-rising population. And this has been confirmed by NASA. Here's a fantastic photograph that they've put together of the Earth. It's a NASA satellite image. And what these colors on the different continents represent is the increase in leaf cover that has happened between 1982 and 2015. And most of this is a green to a dark green color, which shows quite a big percentage increase in the greening of the Earth. It's been as much as 20% since 1982 and 2015. It's been quite remarkable. Now, it's partly because of carbon dioxide and partly because of fertilization that this has happened. But the interesting thing is that the greatest greening has been occurring in the arid regions, which is definitely the carbon dioxide effect. If you look at the Sahara Desert over here, the southern boundary of the desert that you see here is gradually creeping northwards. because as the carbon dioxide levels in the atmosphere increase, so the plants are able to utilize carbon dioxide much more efficiently. They close their stomata, they grow fewer of them, which means they have less water loss, means that plants can start to grow in more arid soils. And as a consequence, we've actually had a net decrease in the Earth's total desert areas. Okay, that brings me, increased CO2 is not only greening the planet, but it's helping to feed it. And here we come to my conclusions, which is that for every breath and every meal and every ecosystem we have, it all traces back to photosynthesis. Photosynthesis really is driving everything that we have. Plants adapt and thrive as CO2 levels rise, and we've seen how they adapted while it was falling. But of course, now that it's rising, they're getting the benefit of some of these adaptations as well. And the Earth is greener today than it was 40 years ago, considerably greener, nearly 20% more. And the final point is photosynthesis remains the quiet miracle that makes our living planet possible. Thank you. I hope you've enjoyed that.
Thank you, Rob. That was fantastic.
All right. Have you got any questions, Tom?
Yeah, my first one is I'm looking at this paper published November 27th. It's called Sunlight, the Bond Albedo, CO2, and Earth's Temperature. And the first author listed is Rob Louw, and then it's William Van Weingarten and William Happer. I think that's a huge honor. I'm extremely impressed. So great.
This paper actually came after my last presentation, where we looked at sunlight, bond albedo, and carbon dioxide. And the purpose of the paper was to see which of those are the three factors that affect Earth's temperature. There are only three that they can be disturbed, but when it comes down to the energy balance, those are the three. And when we did the work, not unexpectedly, the sun's insolence is the biggest one. If that changes, it makes a massive difference. Bond albedo is also big, and carbon dioxide's effect is tiny when you change it. But I leave it for the readers to take a read of it and see what they make of it.
All right. Yeah, that paper is short and sweet. It looks like it's 5 pages, but I'll put a link to that in the show description. And then also I'll put a link to your slides from this presentation in the show description, because there's so much going on, so that people can take a good look at your slides.
Yes. I did pack a lot in. I do agree, but it's quite a complex subject. It's difficult to find this written down anywhere as semi-concisely as I've done it.
One specific question I had is you mentioned maybe towards the middle about stomatal density. And does that change very, very fast? You mentioned, I think there was like a 3-week test that they did.
Okay. Yeah, and I've been asked that before. It's actually not so much that it changes. The plants that they grew over a period of 3 weeks started from scratch. So it was when plants grow in a carbon dioxide rich atmosphere, they immediately start making leaves with a higher stomatal density. It's not that they change after they've grown. Yes, they will do. They'll close some of the stomata if the carbon dioxide levels go up. But it's not an evolutionary process. It happens right away from birth of the plant. So if a plant is grown in an environment which is rich in CO2, it'll only have few stomata. If it's grown in an environment which is poor in CO2, it has to have more stomata in order to get sufficient carbon dioxide through the holes in the leaf. So more holes makes it easier to get carbon dioxide. Fewer holes makes it doesn't need it, and therefore doesn't lose so much moisture.
Okay,
Am
I'm
I
probably, I may not understand it, but if you could take a small oak tree and grow it outside in my yard at 430 parts per million, it would grow a certain number of stomata on its leaves. And then if you could transplant it into a greenhouse where it's 1300 parts per million, maybe the next year when it grew another leaf, then it would already adjust to that higher CO2?
Yeah, I can't answer that definitively, but my guess is that something like that would happen. But let's look at it another way. Tom, if you grow a seedling of any plant and you grow the seedling in a carbon dioxide rich atmosphere and you grow another seedling in a carbon dioxide poor atmosphere, the one that's in the carbon dioxide rich atmosphere will have a lower stomatal density than the other one that is grown. It's a very good point. What happens when new leaves come onto my biology doesn't treat. That's a good. I need to go and check that up. I don't want to give you a definitive answer, but it's quite likely that that's what would happen. That the next time round, the leaves would have a lower density if the carbon dioxide was higher here. I haven't seen research looking at that, but possibly it could be found somewhere.
All right. Any other points you want to make before we finish this one up?
One point, like I made the same point at my last presentation. If there are any U3A societies out there that would like me to do this presentation, I could trim it down a little bit perhaps. Or schools, biology classes, I'm sure that it would be beneficial there, and I'd be more than willing to go in person and do the presentation and take questions. Or any other bodies that are wanting to know a little bit more about the benefits of carbon dioxide, really. So, it gets such negative press per carbon dioxide, yet if you absorb what I've said, you can see it has massive benefits to mankind.
All right. I just want to thank you for doing these presentations, and I'm hopeful that you can do another one maybe in a few months or so. But thank you for doing this. This is really clear, great stuff. So thank you, Rob Louw.
It's been an absolute pleasure. Thank you, Tom.